So we stay with the topic of epigenomics and now have Sara Aibar, postdoc in Stein-Erz lab from University of Leuven, presenting sort of a next step now, combining transcriptome and epigenomics data to actually learn gene regulatory networks. I guess that's sort of an extension of scenic or those ideas potentially. Look forward to that.
Okay. So thank you very much for the introduction. I guess I can now skip this slide and just go ahead and say that in the ARTS lab we are interested in decoding the genomic regulatory code and basically understanding how it come that from a single DNA sequence that we all have inside, so many different cell types emerge. The regulatory programs are driven by these regulatory regions, such as enhancers and promoters, where transcription factors bind by recognizing specific DNA sequences and regulate the expression of their target genes. This association of transcription factors with their target genes is what is represented in gene regulatory networks. Note that in the same way that a transcription factor can regulate multiple genes, different transcription factors can regulate the expression of a single gene depending, for example, on the cell type. In this way, genomic technologies such as RNA-seq and ATAC-seq that allow to profile the transcriptome and chromatin have been very useful to reconstruct gene regulatory networks, specifically in combination with motif enrichment tools. Of course, with the new advances on single-cell technologies, we can now get more resolution and get higher scale regulatory networks. So we have developed a couple of approaches to reconstruct and study these regulatory networks. Since this is a computational conference, rather than presenting a specific study, I will be going and explaining these two methods. That way you will have a better understanding of how they work and hopefully be able to apply it on your own samples or even modify them or get inspired to develop your own methods. In order to infer gene regulatory networks from single-cell and SCOG data, most current methods used to rely on defining a priori either trajectories or cell clustering. But we wondered whether it was possible to infer the gene regulatory networks directly from the data, and whether these networks would could actually be used to detect or infer the cell states. That's why we developed the scenic, which is basically consists on two main steps. One to infer the gene regulatory network, and another one to infer the cell states. In order to infer the regulatory network from the RNA-seq data, what we look is basically for co-expression of transcription factors with potential target genes. Of course, for this we rely on the assumption that the transcription factor and its target genes messenger RNA will co-occur at least in some cells. With this, using co-expression, we get a first draft of the network. However, since this network is only based on co-expression, it does not imply direct binding from the transcription factors, since it can also include many downstream effects. In order to prune this network and get mostly direct targets from the transcription factors, what we use is motif enrichment analysis. We input all the targets of a transcription factor based on co-expression, and then keep only those that have present enrichment of the transcription factor binding motif. This set of a transcription factor with targets is what we call a regulon. By scoring the union of all these the union of all these regulons is what forms the global gene regulatory network. In order to score the activity of the network in each cell, what we do is to take each of these regulons and look at the global expression activity in each cell across the whole regulon. That way we can get like activity distributions and detect those regulons that are likely active in a cell type compared to the regulons that are active in another cell type. This regulon activity score is represented in a matrix where we have the regulons as rows and cells as columns. And here we have this matrix in a real data set. As you can see, we can easily group the cells based on the regulon activity, and these groups of cells mostly match the cell types reported in the original publication. For example, we can get one of these cell types. For example, this little group here is the microglia cells, and we can see that they have a specific gene regulatory network active. Of course, this matrix and this activity can also be used for other kinds of analysis, such as dimensionality reduction, etc. We have regularly applied SCOG to most of the datasets generated in-house, getting similar results. Sorry, I skipped something. Here, of course, we validated that these transcription factors are actually known master regulators of the different cell types, and that indeed applying SCOG is more efficient than just applying, for example, motif discovery on the marker genes of the cell type. Applying SCOG on different datasets, we also came up with some interesting features that it has. The first step that we did was just to confirm that it actually works for dynamic states. So, for example, when we apply it in development, it will reveal the transcription factors leading that development. But actually, the most striking results came from the fact that the regulon activity matrix is somehow a biologically driven dimensionality reduction. So, for example, in cancer, when we try to cluster the cells directly, they normally tend to cluster, for example, by patient of origin, the color in this TSNE. However, when we apply SCOG, we get more biologically relevant features highlighted, for example, in this case, the cancer subtype. This robustness to batch effects and technical bias is especially useful, and we went to a bit more exotic analysis, such as comparing the regulatory networks across species, for example, from mouse and human brain cells. This is not only, we didn't just compare the actual targets of each transcription factors, but we also compare whether the regulons actually represent the same cell types across the different organisms. And actually, this contrast is robust enough that it actually allowed to cluster the cells from the same type even across organisms. As I said before, we have regularly applied SCOG to most of the data sets that we have generated in-house, getting similar results. For example, here we have the results on the fly brain single-cell NSEC dataset, where we have basically coverage of most of the cell types in the brain of the fly. And SCOG efficiently recovered the regulatory networks underlying the major glia and neuronal cell types. But it also revealed an otherwise hidden axis that it's related to oxidative phosphorylation. In addition, lately we have also developed a new tool, SCOG, which allows to explore the results from SCOG, or actually any LUM file that you input into it. It's available at this URL, but it can also be installed locally. In the main tab, you can just explore the activity of specific genes. For example, we have ILES and Portabella representing the canyon cells in the fly brain. But in this other tab, you can explore the results from SCOG. For example, a regulon representing glial cell types and a regulon representing the canyon cells. Here we have the regulon activity and the binarized regulon activity, which allows also to compare it directly to the transcription factor expression. This is especially useful because many transcription factors are expressed at low levels, and therefore they are very prone to dropouts. So until here, I have shown that it's actually possible to infer gene regulatory networks from single-cell and NSEC data, but actually epigenomic techniques are more prone to this task, since they provide the regulatory regions that are actually active, and therefore they are more adequate to try to reverse engineering the regulatory networks. In particular, single-cell ATAC-seq, what it provides is the regions that are accessible, for example, because a transcription factor has displaced the nucleosomes, and in bulk it provides a profile similar to this, where we can easily find the regions that are more accessible, unlikely the regulatory regions providing the cell its identity. However, in single-cell level, this data is normally very sparse. Actually, for each cell, we normally can sample only a subset of the accessible regions. So it's not possible to actually, it's not possible for a specific cell neither to call the specific, to determine whether a specific, to call a specific region, not to say whether that specific region is accessible in that specific cell or not. We need different specific statistical approaches to actually try to infer this information. The two main approaches are basically region-based, where you collect based on motifs or non-regulatory regions, aggregate all the reads in the cell across those regions, and then use those aggregates of regions and cells to do cell clustering, or the reverse approach, in which you initially cluster the cells and then aggregate all the cells from the same cluster to do a pseudo-bulk and do a differential accessibility analysis across them. While these approaches have been shown satisfactory to detect the cell types, it hinders the discovery of new regulatory programs. So that's why we also developed Cystopic, which is a method based on LDA. As we saw yesterday, this is a technique commonly used in text mining. And it basically allows to infer two probability distributions. One that relates the cells to the topics, and one the topics to the regions. Note that each cell and region can be associated to multiple topics. These probability distributions can be used to either cell clustering or enhanced cell clustering, which would basically represent the different regulatory programs. We did extensive benchmark of this topic to actually confirm that the cells and the associated regulatory regions are correct. And basically, some of the interesting features that we found in this case is that this topic is very robust, especially at low coverages, which is actually for low coverages, for example, like the ones coming from combinatorial indexing methods. We also found that optimizing the cell and enhancer clustering is actually especially useful for dynamic processes, for example, like in differentiation. The fact that the cells are distributed along a trajectory and they are cannot be assigned to the distinct clusters is actually a problem for methods based on clustering or aggregation, but not for Cystopic because it just uses probability rankings. So here we have the results of Cystopic applied to a set of hematopoietic differentiation. And here we can see the aggregate of all the cells for a given cell type across the different topics, having here representative regions for each topic, where this region was selected within the top of the probabilities of being open within that topic. We can see that there are some specific topics that are generally accessible and mostly represent proximal promoters, but some others are more branch-specific or cell type-specific. These probability distributions can also be used to reconstruct the differential trajectory or to cluster the regions and explore the underlying regulatory code. In addition, these topics or sets of regions can also be used for many other types of analysis. For example, I will illustrate here the cross-species network comparison with these data sets. We applied Cystopic to single-cell ATAC-seq data sets from human and mouse brains, and we easily recovered the cell types that they had been reported in the publications, but really far more easily. And then we compared the human topics to the mouse topics using enrichment and just doing the corresponding regions using leftover. So we see that actually the cell types that present the most conservation across species are the glial cell types. In the same way, we also compare the human topics to the results of applying Cynic on a similar single-cell NSEC data set, and it was nice to confirm that actually the topics and the regulons agree to a great extent, especially in regards to transcription factors and the related binding motifs. So the things that, for example, we could not have done with single-cell RNA-seq is something like this. Since the topics provide the regulatory regions, it's also possible to do some further analysis. For example, this represents all the regions that belongs to the oligodendrocyte topic in human, and then also all the regions from the oligodendrocyte topic in mouse. We can get some of the corresponding ones using leftover, and see that actually the enhancer architecture is really conserved in some of them, keeping even not only the same motifs, but even the relative structure position across them. So to finish, just to mention that Cynic and Cystopic are both implemented in R and available in GitHub. You have all the links in the Arts lab. But for example, Cynic was very slow to run, especially because the co-expression network for big data sets was really slow. So we re-implemented the co-expression network in Python into GRM Boost using gradient boosting machines, and now we have re-implementing the whole pipeline in Python, so it's very easy and fast to run, even for big data sets in just a few minutes. However, as you can guess from this slide, installing all these packages and keeping track of the dependencies and versions, it was a nightmare. So we have also recently packed it into containers from Docker on Singularity. Actually, this is the work of Chris, which is actually in the audience. And merge all these Dockers can also be integrated into Nextflow workflows to actually do the analysis much more easily. For example, running Cynic or any tool in batch to multiple data sets, which is especially useful, for example, for meta-studies or to apply it to cell atlases. And it can also allow the easy integration with other tools, like ScanPy, for example. And in the future, we probably plan to do this also for SysTopic. So in summary, I have presented Cynic and SysTopic, which both allow to simultaneously map gene regulatory networks and cell states using either RNA-seq or single-cell epigenomics data. And the main conclusion is also that they provide concordant but complementary gene regulatory networks to study the transcriptional regulatory code. So thank you for all the people involved in the project, which is in these projects, which is actually the work for most of the people in the Stein Arts Lab. And thank you, of course, also to all the founding and you for listening. I hope it was useful.
Thanks, Sara. That was a tour de force through many tools that you guys are doing, very exciting. And thanks for giving us an overview. We have time for a few questions. Sam?
I just didn't quite follow if SysTopic is LDA that you've shown works well in single cell, or if there's something beyond the basic LDA implementation that is associated with that.
It's mostly LDA, just using the cells as document and the regions as words. I think that's the order. And we just used a collapse scheme sampler at the beginning to actually define all the parameters that are required to the probability distributions, but that's basically it.
So I would assume that like an NMF-based or sort of similar approach would potentially not, I mean, you need a good latent space essentially, right?
Pardon, what do you
mean? I would assume we talked the same thing, a non-negative matrix factorization-based latent space generation or some other thing could, I mean, I think you did beautiful validation, right? I'm just not seeing why this would be so specific to this thing. So I think it would be nice potentially to follow this up. Is your validation, is it easily usable to try around with other latent space generation methods?
Yeah, actually I'm not involved in the actual method, development of the method, so I don't really know how to apply this latent space validation. But I can get you in contact maybe with Carmen, who developed the method. I really cannot reply this question.
It's not I person, but I mean, there's people interested in that, so I think it might be a nice community effort. Maybe a hackathon thing next time.
Actually, if you want, related to the questions that we had before, for example, in regards to choosing the peaks or the input features for the matrix, I would like to mention that I have lately been using SysTopic mostly as a user. Okay. And actually, the input of the regions doesn't matter that much. It's better to have, for example, a lot of extra regions that have very low coverage, and SysTopic will just not use them. So normally what we use is just either all the regulatory regions across the genome, even if they are not used in our specific cell types, or just call the peaks with a very lowly stringent cutoff, so we just can get peaks representing all the cell types. And whatever is not relevant, it will just not be chosen and not assigned with high probability to the topics.
That makes sense. I was interested in the fact that you use Nextflow and sort of package things into a Docker container. So this is trying to make things essentially ready for some type of core facility type of processing, right? Which obviously doesn't want to have any interaction or sort of parameter setting. Mm-hmm. So I haven't seen this so often happening, even on the transcriptomics side. So what's your take on the field? Are we ready to sort of really package things away, go to default parameters, and sort of just work on the outputs, which is essentially what you're going for here? Yeah.
Well, actually, I can talk to you from experience, for example, with Cynic. Initially, we developed it just as a pipeline where we just do the different steps manually and checking the parameters and such. But after applying it to different data sets, we just saw that most people, even ourselves, we just end up using exactly the same parameters for everything, and that they tend to work at least reasonably well across them. So I think it's really not such a crazy idea to just have a pipeline with set of defined parameters to work with it. Also, it's true that Nextflow is probably more oriented towards, for example, when you want to do a meta-study or when you want to apply specifically the same pipeline to multiple data sets. If you just want to apply it to one, maybe just with the Dockers that are also a bit more interactive is already enough. But certainly having the Dockers helps a lot because otherwise, yeah, dealing with the different versions is really a mess.
Thanks again for a nice presentation, Sara.
Okay, thank you very much.